Law. Okay, very good morning, my dear friends. So, in the regarding of this CPGET 2023 free online coaching for all competitive exams, also in plant sciences, we are going to give free online coaching through online mode through Zoom platform. So, today our eminent resource person, Dr.A.Kavitha Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College, Autonomous, Hyderabad. Madam is going to explain about the Pteridophyta. Yesterday, Madam has clearly explained about the Lycopodium type study and life cycle of the Lycopodium. So, remaining, we're going to explain today onwards, my dear friends. Listen carefully during the class. If you have any doubt, please you clarify your doubts with our respected Madam. Please welcome, Madam.
Okay, sir.
And share your screen and start your session.
Right, sir. Thank you. Good morning, participants. Today we are going to start with the next type study of Pteridophytes, that is Equisitum. So just I'm start sharing my screen. So this is yeah. Yeah, this is the Equisitum, the next type study of the Pteridophyte, which are commonly known as the horsetails. Now, next we're moving on to the characteristic features of this Equisitum. So it is popularly known as the horsetails. Why it is called as horsetails? Because you can observe here all the leaves. This is the central part of the central axis, and the leaves are just like the pointed leaves. Okay, so the leaves will be in the form of jointed, like a limb, like a bone. How the joints will be, the leaves will be in that manner. Or we are calling it also as a scoring brush, like the brush with which what we'll clean the bottles and all that. It appears just like that. Now, it is represented by around 30 species worldwide, except in Australia and New Zealand. So in these two countries, we couldn't see the growth of this Equisitum. And remaining all the regions of the world, we can see the presence of these Equisitum species. And they prefer these species, they prefer damp and shady places, whereas majority of them, they are growing in marshes, ponds, or the streams. Most important one is they are also found in xerophytic habitat also. Now, some common Indian species includes Equisitum arvens, Equisitum debile, elongatum, Equisitum diffusum, Equisitum dubium, etc. Now, so this is another important point to be remembered here. Equisitum, these are considered as the indicators of the minerals in the soil. That means to say, if the soil is going to consist of some minerals, like a huge quantity of minerals are located in that particular area, there will be the more growth of this Equisitum in that, which indicates that we can mine the minerals present in that soil. Especially, they can absorb the gold from the soil. So this is a very important indicator. Now, so the deposition of the silica on the outer wall of the epidermal cells, it makes the plant appears to be rough in texture. Okay, so the plant will be very much tough, or it will be very brittle also, we can call it as, by the deposition of the silica. So they are always used as the abrasives. So due to this property, they are always used as abrasives because they are very rough in structure. So anything which we want to clean or to rub something, we can use this. So it acts like an abrasive also. And the silica which is deposited on the plant body, it is acting like a protective covering against the pathogens. And some of these species, they are also used in the Ayurvedic medicines as diuretics also. So that is some important characteristic features of the Equisitum. Now we are moving on to the external morphology of the plant body, that is a sporophyte. So the plant body is divided into stem, root, and the leaves. Now, it consists of a perennial, prostrate, branched, and creeping underground rhizome. Now, if you see the picture here, this is the underground, prostrate, and creeping rhizome. Prostrate, that means to say, and it is perennial. Okay, it is attached to the soil. It will be creeping and it will be present underground the soil and the creeping. And it will be branched also.
Madam, excuse me.
Now,
Good morning,
yes?
madam.
Good morning. Okay, shall I
Yeah,
continue?
ma'am, continue. Start it.
Right. So the equisitum, it is going to consist of a perennial, prostrate, branched, and creeping underground rhizome. Okay. So from which we can see the development of these aerial branches. Okay. So this is the rhizome part of the plant body, and this is the upright branches, which we call it as the aerial shoot. Now, aerial annual branches. So some of these aerial branches, they are sterile and some of them are fertile. So why we call them as sterile and fertile? Because the fertile branches, they are going to develop the strobili, whereas the sterile branches, they doesn't form any strobili. Now, so these aerial branches, they are usually thin and they are vine-like. That is, they are the stem is very weak. It will be climbing. It will need a support to climb. So that's why we're calling it as vine-like. And they climb over the adjacent trees and it is herbaceous. Okay, right. Now the stem, it is represented by branched and creeping perennial underground rhizome. It is divided into nodes and internodes. And at each node, you can see the presence of small scaly leaves. So you can see here, this is the nodal region. These are the nodes. At each node, we can see the presence of some scaly leaves. Now, at the axil of each scale, there is a branch, but so they develop into an aerial or underground branch. Now, in some species, at this nodal part, it may also form some small and round tubers. So, which when they separate it from the rhizome, it develops into a new plant. You can look into this here. This is a nodal part of the rhizome, and this is the tubers. Okay, and extra outgrowths. So they are round and short enough. They help in the vegetative propagation in developing a new plant. So this is the whole structure of a Equisitum plant. This will become the rhizome, which is also differentiated into nodes and internodes. And from the nodal region, this one is the aerial shoot, which is also divided into nodes and internodes. The nodes are going to develop some sterile branches, or we can see the presence of the scale leaves also at each node. And the fertile branches, they develop into the strobili or the cones at the time of reproduction. So this we have done. Now next, moving on to now external features. Is herbaceous. Right. So the rhizome or the stem, both the rhizome as well as the stem, it's going to consist of two types of shoots. That is the aerial shoots, or we can even call it as vegetative shoots or the fertile shoots, which are branched. These aerial shoots or the fertile shoots, they are branched. They are greenish in color and perform photosynthesis. And the fertile shoots, they are unbranched without any chlorophyll. And each of them, they terminates in a cone or the strobilus. So at the time of reproduction, so this is a fertile shoot, which is unbranched. You can see here, this is not branched. Okay, it is an unbranched shoot. It is an unbranched shoot without any chlorophyll, and they will terminating with a cone or the strobilus at the typical part. Now, the number of these branches. So the very important point here to be noted is in Equisitum, the number of the branches will be equal to the number of the leaves at the node. Suppose at the node, it is developing 2 branches. So the leaves which are present at that branches will be 2 in number. Number of the leaves is, this is again an important point to be noted. Number of branches will be equal to number of the leaves at each node. Okay, so you can look into this here. At each node, how many leaves will be there? So many number of branches it will be going to develop. Now, so these branches, whatever are going to develop at each node, these are of 2 types. One is called as branches of unlimited growth and the branches of the limited growth. That means to say, some of the branches, they are growing unlimitedly and does not forming any cone or the shrubulus. Whereas certain branches, they are limited. They grow to a certain height and they'll stop their growth. Those are called as the limited growth branches. So this is the nodal region. Okay, and these are the scale leaves. And this is the strobilus. At the time of reproduction, you can see this is a cone or the strobilus. Now, so the branches of the unlimited growth, these are similar to the vegetative shoot. They grow unlimitedly and divided into nodes and the internodes. Whereas the branches of limited growth, they have only a limited growth. They grow only up to a certain period of time. And they are unbranched. So these branches of limited growth, they are unbranched. And due to this, rhizome and the aerial shoots, they have jointed appearance. So the internodes are very hollow and having the longitudinal ridges and the grooves. So this in the internal structure, I'll tell you. The ridges of the successive internodes, they alternate with each other. And number of ridges will be also equal to the number of the leaves at the node. Now, when I talk about the leaves are minute, very, very small. They are scaly and isophilous type. That is, the dorsal side and the ventral side, both the sides of the leaf is same. Next, they are arising in whorls at the nodes of both underground rhizome as well as the aerial branches. Now, these leaves, they fuse at their base to form a base sheath, a distinct sheath, and their free distal ends give a frill-like appearance. So just now I'll show you here, right? So these are the leaves. Okay, so these leaves, they are all joined or they are attached with one another at the base. And whereas the upper part of this leaf, it is forming a frill-like appearance. Now the leaves have only a single midvein in the center. Chlorophyll is absent, hence they are non-photosynthetic. But the main function is to protect the branch buds at the nodal region. Now the photosynthesis, now the leaves here does not have any chlorophyll. Then where the photosynthesis, how the photosynthesis is occurring in Equisitum? It is carried out by green and the sterile branches of the aerial part of the rhizome that will be performing the photosynthesis. Now, when you talk about the roots, all the roots of the Equisitum, they are first or primary root of the embryo, that is the adventitious, but later on they become slender and fibrous and they arise from the basal of the node or the rhizome or from the base of the upright branches. So you can look into this here, the structure. These are the roots which always develop from the base of the nodal region of the rhizome part. And the roots are of adventitious type or fibrous. Now, so they are branched and they branches, they are endogenously developed from the pericycle. So this from the internal anatomy, I'll tell you what is this. So these roots are the branch. Now when we go for the anatomy of the stem here, so this is a very important part to be remembered in the anatomy of the stem. So the anatomy of this stem, it shows both hydrophytic as well as the xerophytic characters. Now, when we talk about the anatomy, here the outline of this tiers of the stem, it is wavy in outline. You can look into this. This is a wavy in outline due to the presence of ridges and furrows. These are the ridges and these are the furrows. Okay. The stem will be in the form of wavy outline, giving rise to the ridges and the furrows. And it is going to have a distinct epidermis with well-developed cortex and a stele and with a ring of vascular bundles and a central large pith region. So the outer epidermal region, a well-developed epidermal region. This is the central, the middle cortex, and this is a stellar region represented by a ring of vascular bundles, and this is the central hollow pith. Now, so this is now. So when we talk about the characteristic features here, just by looking at this, I can explain you here. So below the each ridge, you can see the presence of sclerenchymatous, that is a xerophytic origin, xerophytic character, presence of the sclerenchymatous tissue. And in the cortex region, you can see here, these are all are the air chambers. Okay, some empty spaces which are present in the cortex region. We call them as the vallecular canal. This is very, very important point. Vallecular canals are present in the cortical region of the equisitum stem, which imitates, or we can say that this is a characteristic feature of an hydrophyte. So in all the hydrophytes, that is in all the aquatic plants, we can see the presence of the aerenchymatous tissue. That is, the tissues are present with air cavities or the air chambers. So the same characteristic feature appears here also. So that becomes an hydrophytic character, and a central pith region, an empty central pith region, and the presence of this sclerenchymatous tissue and deposition of the silica over the epidermal region, it gives rise to the xerophytic character. So equisitum stem shows anatomically, shows the xerophytic characters, and at the same time hydrophytic characters also. Now we can see in the stellar region, that is in the having a single layer of endodermis and the pericycle, and these are the ring of vascular bundles here. And you can look into this here. These vascular bundles, below the vascular bundle, you can see a small cavity again. Each vascular bundle is attached with a small cavity or a small opening. We call it as the carinal canal. So presence of vallecular canal and presence of this carinal canal is the distinguishing identification characters of equisitum stem. Right, you can look into this. Okay, the cuticle, this is the sclerenchymatous tissue. We call it as hypodermal tissue. This is the vallecular canal. This is the phloem, and these are the xylem, protoxylem here, and the xylem, and this is a carinal canal. Okay, so we'll go with the description again. The epidermis here, it is uniciliate, a single layer with a heavy silica deposition on the outer wall. The epidermis is interrupted with sunken stomata. This is xerophytic character. The stomatal apparatus, it is going to consist of 4 cells arranged in 2 tiers, and a series of siliceous thickening band that develop on the lower surface of the accessory cells. Next, cortex is differentiated into outer and the inner. The outer cortex here, it is sclerenchymatous hypodermis, and it is multi-layered. Below the ridges of 1 to 2 layered, and in the grooves it is only 1 to 2 layered. Now below this stomata there is no sclerenchyma. So sclerenchyma is followed by a band of sclerenchyma performing photosynthesis, and inner cortex is mainly made up of thin-walled parenchyma cells. Beneath the floor, you can see the presence of a large schizolysogenous cavities, or the canals. We call them as the vallecular canals. So these are continuous in the internal portion, and they form a distinct aerating system. So this is the characteristic feature of aquatic or the hydrophytic character. Now in between the stele and the cortex, there is a single layer lying, which we call it as endodermis, followed by a single layer of parenchymatous pericycle. Now when we go for the vascular cylinder, it is of siphonostele, in which the vascular bundles are arranged in a ring around the larger pith region. So the number of vascular bundles is equal to the number of ridges and also the number of the leaves. So all these will lie in the same straight line. Number of leaves, number of number of ridges, and the number of vascular bundles, they are equal in number. So this point మీరు గుర్తుపెట్టుకోవాలి. In Equisitum, number of leaves, number of ridges, and number of vascular bundles, they will be equal in number. Now, these bundles, they will alternate with the vascular canal. Now, the vascular bundles are considered to be as the conjoint and collateral and endarth. So that means to say, both the xylem and the phloem, they lie on the, they lie together. We call them as conjoint and collateral. Cambium is absent. As I told you, these are the primitive vascular cryptogams. And phloem is well developed, whereas xylem is poorly developed. Protoxylem is consisting of annular or spiral tracheids. And metaxylem, it is made up of pitted tracheids. Now, in mature vascular bundles, the protoxylem is going to degenerate or disintegrate to form a lacuna or an empty space, which is called as carinal canal. So the carinal canal, it forms an inner ring of the intercellular spaces, which is filled with probably the water and helps in the conduction of the water from one place to, from one part of the tissue to the another part. Now, the xerophytic characters, I told you, Equisitum is showing the xerophytic characters. What are those? Presence of ridges and furrows, the epidermal cells with silica deposition, sunken stomata, sclerenchymatous hypodermis, reduced scale leaves, photosynthetic stem, and a well-developed vascular cylinder. So they all comes under the xerophytic characters of the Equisitum. Now, what is the hydrophytic features? Presence of a well-developed aerating system, what they call as vallecular canals, and the presence of reduced xylem. So that becomes a hydrophytic features. So this is also the same thing. Now, when you go for the anatomy of the rhizome here, anatomy of the root. So you can look into this when we talk about the anatomy of the root. It has epidermis, cortex, and the stele. The root epidermis it is uniseriate with thin-walled cells and protective in function. And from this epidermal hairs will arise, which are elongated, and the cortex is differentiated into sclerenchymatous outer cortex and the parenchymatous internal cortex. So when we go for the stellar region, the stellar region is surrounded by a single layer of endodermis and pericycle. Internally, we can see the xylem and the phloem, which are right. So xylem and the phloem here. The xylem and the phloem, it is going to form a protostelic condition with a diarch or hexarch xylem. So that means the number of the xylem, they are 2 or 4 or 6 in number. The number varies from one species to the another species. Now, a large metaxylem tracheid is present in the center and a protoxylem. So this is a large metaxylem in the center surrounded by the protoxylem. Now, phloem bundles, they are going to alternate with the xylem strands, and pith is going to be absent. There is no pith. This is the phloem strands that alternating with the xylem. This is about the vegetative structure or the external morphology of the Equisitum sporophyte. Now, when I'm moving on to the reproduction point of view, the vegetative reproduction of Equisitum, it is carried out with the help of the rhizome which is present underground, or with the help of the tubers, which will be developing on the rhizome at the nodal part. Now, it can next part, the reproduction is also carried out with the help of the spores. Now, where these spores are developed within the sporangia, and the sporangia, they are born on the sporangiophores, which are aggregated to form a compact structure, what we call it as strobulus or a cone. Now, you can look into this structure here when I show you. Right. So this is the strobulus or the cone. The sporangia, they are developed on a small stalk. We call it as sparaginous stalk. And all them, they are aggregated together to form a cone or the strobili. Now, when we talk about the structure of the strobili here, the strobilus, it is the terminal position which are borne terminally on a chlorophyllous vegetative shoot. I repeat, this is an important point. The position of the strobilus in Equisitum, it is borne terminally at the tip region of the chlorophyllous vegetative shoot. Now, the strobilus is going to consist of a whorl of sporangiophores. That is the sporangia, they are developed on a stalk. So that stalk is called as sporangiophores. A group of these sporangiophores will form the strobili. Now, each sporangiophore, it is a stalked structure bearing a hexagonal peltate disc at a distal end. And on the undersurface of this sporangiophore, disc 5 to 10 elongated cylindrical hanging sporangia are born near the periphery in the form of a ring. Okay, so this forms the structure of the strobili. The flattened tips of the sporangiophores, they fit closely each other and provide the protection and developing the sporangia. Now, how is the sporangia? The mode of development of sporangia here is eusporangiate type. Okay, so it is not entirely formed from a single initial, but the superficial cells will also form or take part in the development of the sporangium. That type of development, we call it as eusporangiate type of development. Right. So here the sporangia, how are they? They are initiated in the superficial cell around the rim of the young sporangiophore. The sporangium initial forms an inner and the outer cell. Inner cell, by further division, they gives rise to the sporogenous tissue. The innermost layer of the sporangial wall, it takes place as a function of the tapetum, and the sporogenous cells, they separate from each other and they transform into the spore mother cell. Now, these spore mother cells, they get nourishment from the tapetum, the inner lining of the sporangium, and each mother cell, it will undergo meiotic division and produces a spore tetrad. And all the spores which are developed in the sporangium, they are of same size and shape. Hence, Equisitum is also regarded to be as the homosporous type of development. So the mature sporangium, it is elongated sac-like stalked structure attached to the peltate disc of the sporangiophore, and it is surrounded by the jacket loop. Now, how are the spores? So these spores they are spherical in structure. They are filled with densely packed food material and the chloroplast, having the innermost layer called as intine, followed by the outer thick exine. Now, these spores, they are liberated out from the sporangium and from the strobilus, and they fall on the substratum and gives rise to the next generation that we call it as the gametophytic generation. Now, we are moving on to the sexual reproduction. That is, the spores upon germination, they develop into the gametophyte. Now, the gametophyte here, the gametophyte of Equisitum plant, it is a monoecious species. That means the male sexual organ, that is antheridium, and the archegonium, they are developing on the same gametophyte. And now here, antheridia are the archegonia are the one first developed, later are the archegonium. Now the antheridia, they first appear on the lobes of the gametophyte, and the periclinal division of the antheridial initial, it gives rise to androgonial cell. So I'll show you here the development. So this is the development of the antheridium, and this is the development of the archegonium. So this is the antheridial initial, which is developed on the gametophyte. So one of the superficial cell of the gametophyte will get transformed as the antheridial initial. So this antheridial initial, it divides by one anticlinal division and going to form one androgonial cell and the jacket initial. The androgonial cell upon division and transforms into androgonial cells. And even the jacket cells are also dividing, and it will surround this developing androgonial cells for giving protection. Now, each androgonial cell of this tissue will transform into androcyte mother cell, and each androcyte mother cell will divide and develops into the male gametes, what we call it as the antherozoids. So these are the antherozoids. So one peculiar characteristic feature of these antherozoids are the antherozoids here are spherically coiled. Okay, basically the upper part of the antherozoids, they are spherically coiled, and the lower part is expanded. So you can look into this. This is the upper part of the antherozoid, which is spirally coiled, just like a shell of a snail. How the shell will be, the upper part of the shell of the snail will be coiled. It appears just like that. And these are the multiciliated flagella. That means the flagella are many in number at the anterior position. And the lower part of this antherozoid is expanded. Okay, so the antherozoids which are developed in the equisitum, they are spirally coiled and multiflagellated or multiciliate. Now, when we go for the archegonial development, so just like that, within the gametophytic tissue, one of the cell will transform as the archegonial initial. So this archegonial initial, first it will undergo one division and form one primary cover cell and the central cell. The primary cover cells will transform into the next cells, and the central cell will behave like, or it will undergo one more periclinal division and forms ventral canal cell and the primary neck canal cell. Now this ventral canal cell will divide further. This ventral cell will divide further and forming ventral canal cell and the female gamete egg, and the neck canal cells will divide. And here one more important point is, in equisitum, the archegonia, the neck is very, very small, representing by only 2 neck canal cells, and these are the 4 neck cells. So this is regarding the archegonia, which is embedded inside the gametophytic tissue of the equisitum gametophyte. Now, so fertilization. So here also water plays a very important role here for fertilization. So this gametophyte, it must be covered with a thin layer of water in which the motile manthrozoid, they swim and reach the archegonia. Now from the antherozoids, when they reach the neck canal cells and the ventral canal cells, they will degenerate and it will create a passage for the entry of the antherozoids into the venter. And then it is these antherozoids, they are fusing with the egg and forming the diploid zygote. Now this zygote, upon germination, the zygote will develop into the new sporophyte, what we call it as the embryo. So embryo, it is a mother cell for the next generation, that is the sporophytic generation. Now this embryo, it will undergo differentiation and develops into the and it will develops into a new equisitum or a new adult plant. So this is how the development of the equisitum takes place. The embryo first undergoes a quadrant division, followed by a multicellular and multinuclear division, and results in this young sporophyte again. Okay, this is the gametophytic tissue, and this becomes a young sporophyte stems. So the life cycle here we can see. So this is a sporophyte, the equisitum plant. So this is a fertile branch. Okay, this is the fertile branch developing the strobili or the cone at the terminal end. So this is the strobili or the cone. So you can see here on the strobilus, we can see the presence of sporangiophore. these sporangiophores, how they are arranged? In a ring around the around the strobili. So it will be having this sporangiophore is going to consist of a basal stalk, and this is a peltate disc. Now below this disc, these are the sporangia which found to be hanging below the disc. Now, so these are the sporophyte, the sporangium. Now from within the sporangium, the sporogenous tissue will undergo meiosis division and results in the spores. So these are the spores which are released from the sporangium. Now these spores, they are developing into the new prothallus, what we call it as a gametophytic tissue. Now within this prothallus, at the terminal tips, it will develop the antheridium and also the archegonium. Now within the antheridium, they will form the anthrozoids, which are multiciliated or multiflagellated, and this is a female gamete egg. These two gametes, they are undergoing fusion and resulting in the form of the zygote. Now this zygote will transform into the young sporophyte, what we call it as embryo. Now the embryonal differentiation will occur and results in the formation of the new sporophyte. So here you can see the diagrammatic representation. How is the sporangiophore? This is the stalk of the sporangiophore, the axis, and the peltate disc, and these are the sporangia attached to it, a ring of sporangia. These are the spores. This is the male gametophyte and the female gametophyte. These are the anthrozoids. You can see the spirally, apical part is spirally coiled, and the broad basal region, zygote developing into the embryo, and a young sporophyte again. So this is regarding the Equisitum type study. So any doubts in this? The main important points to be remembered here in Equisitum is regarding the anatomy of the stem, Equisitum stem, presence of vallicular canals, presence of the carinal canal, presence of the central pith region, and possessing xerophytic characters of sclerenchymatous hypodermis, the cuticular epidermal region, and development of the aquatic characters of the presence of air chambers. Okay, all that should be remembered for the Equisitum stem. And in the reproduction point of view, the anthrozoids in Equisitum, they are spirally coiled at the terminal part, and they are multiflagellated or multiciliated. So these are the points to be remembered. So any doubts in this? No doubts? Okay, right. So if you are not having any doubts, so thank you so much. We'll meet tomorrow with another type study of the pteridophyte, that is Marsilia. Right. Okay then, thank you so much. Raji, my recording stop cheyochu. Right. Thank you so much now. I leave. Tomorrow we'll meet again. Thank you.
Sir. Sir. Sir.